Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Central University's Common Entrance Test Exam question paper conducted in the year 25th June 2023. This is Biochemistry question paper that I am explaining. If you are new to my channel, please do like, share and subscribe. I have also uploaded previous year question papers of Biochemistry which was conducted by CUET. You can refer to them in my channel playlist for the same. Coming to the first question. Arrange the following steps of cell signaling in chronological order. Reception, generation of signal, response, transduction. So signaling pathway, it involves generation of signal. First the signal has to be generated. Then reception of the signal, then transduction of the signal, then response. So the answer here is option 1, that is BADC. Then given below are two statements. Statement 1. Histone acetylation is generally associated with a decrease in gene transcription. No. That is incorrect. Histone acetylation, it is associated with increase in gene transcription, not decrease, because acetylation, it relaxes the chromatin structure, and as a result of that it becomes more accessible to transcription factors and other regulatory proteins, thereby increasing gene expression. So first statement is wrong. Then acetylation occurs on lysine residues in the N-terminal tails of histone. That is correct. So statement 1 is incorrect, but statement 2 is correct. Answer is option 4. Then next one. Mutation in the middle of a gene that creates stop codon is called frame shift mutation. It is not frame shift mutation, it is nonsense mutation. And frame shift mutation occurs when there is a change in the reading frame of the reading frame by insertion or deletion of one or more nucleotides. Frame shift mutation can be a point mutation. That is correct. So here also statement 1 is false, but statement 2 is true. Then next one. Weak interaction like hydrogen bonds and salt bridges stabilize protein folding. That's correct. Protein folding is assisted by chaperones. So both statement 1 and 2 are correct. So answer is option 1. Then enzymes lower activation energy of reaction. That's correct. The presence of enzyme has no effect on K equilibrium constant. That's also correct. So both statement 1 and 2 are true. Answer is option 1. Then 146 base pair of DNA are wrapped around the histone octamer to form a nucleosome. That's correct. Nucleosome play an important role in gene expression. That's also correct. So both statement 1 and 2 are correct. Answer is option 1. Then ammonia is produced during catabolism of amino acid in muscle cell. That's correct. In muscle cells, ammonia is converted to urea and then excreted through kidney. That's not correct. Ammonia is converted to glutamine, which is then transported to liver for conversion into urea and finally excreted through the kidneys. That is more correct. Not muscle cells, ammonia is converted to urea. It is in the liver that ammonia is converted to urea. So statement 1 is true, but statement 2 is false. Answer is option 3. Then cyanide result in cellular hypoxia. What is hypoxia? Hypoxia is a condition when the body or part of a body does not receive sufficient amount of oxygen. That is hypoxia. So first one is correct. Statement 1 is correct. Statement 2 is the tight coupling of electron transport and phosphorylation is uncoupled by cyanide. It is not an uncoupler, it is an inhibitor of complex 4, cyanide. So statement 2 is not correct. So statement 1 is true. So the answer here is option 3. Statement 1 is true, but statement 2 is false. Then next one. What is true about steroid hormones? Steroid hormones bind to specific receptors. Yes, that's correct. Steroid hormones bind to DNA to control, they don't directly bind to DNA to control gene expression. So steroid hormones, what happens is that steroid hormone, they diffuse across the cell membrane, bind to specific receptors in the cytoplasm or nucleus. Then they will cause a conformational change in the receptor. Then the receptor hormone complex, it will translocate to the nucleus and bind to specific DNA sequence called as hormone response elements and thereby regulate gene expression. So it doesn't directly bind to DNA, it is the receptor hormone complex that bind to hormone response elements present in the DNA. So B statement is not correct. Steroid hormones travel to distant tissues through blood. That is also correct. So the statements which are correct are A and C only. Answer is option 4. Then which of the following statement is true about histones? Histones are rich in acidic amino acid. No, it is in basic amino acid. So B statement is correct. Histones are important for stabilizing single-stranded DNA. It is double-stranded DNA, not single-stranded. Histone binds to DNA to form chromatin. That's also correct. So B and D are the only correct statements. Answer is option 1. Then resting membrane potential is conducted by, so active transport by Na plus K plus ATPase, which pump Na positive out, K positive in, creating concentration gradient. Then selective permeability of phospholipid bilayer, that is also correct. Passive diffusion across lipid bilayer, no. It is differential distribution of ion across the membrane, resulting in membrane potential, creating membrane potential. So A is correct, B is correct, and D is correct. So A, B, D, the answer is option 1. Then next one. Which of the following proteins do not belong to the group of chromoproteins? Fibroin. Fibroin do not belong. They have asked do not belong, not. So it is fibroin. Hemoglobin is a chromoprotein. Keratin again is not a chromoprotein. Cytochromes are, flavoproteins are. So A and C only. Answer is option 2. Then next one. What information about protein can be derived most accurately from gene sequence? So gene sequence, if you have, you can convert it into amino acid sequence. From amino acid sequence, you can determine the primary structure of the protein. So A only will be the answer. Function of protein, secondary structure of protein, three-dimensional structure of protein, all these cannot be accurately determined. Then two proteins, molecular weight 20 kDa, Pi 6.3 and Y, molecular weight 90 kDa and Pi 6.4 can be separated by which of the following techniques? Gel filtration, yes, they have different molecular weight. So gel filtration can be adopted. Ion exchange chromatography cannot be used. Isoelectric focusing also cannot be used because Pi is almost same. SDS page again can be used because they have different molecular weight. So A and D only. Answer is option 2. Then next one. Which of the following techniques can be used for the analysis of gene expression? So DNA northern blotting, yes. RNA sequences are, RNA is isolated and then blotted. Southern blotting, it is for DNA, so B cannot be used. Western blotting for proteins, of course they can be used. This gene expression. Then QRT-PCR, yes, that also can be used, and RNA sequence, that also can be used. So A, C, D, E only. Answer is option 2. Then comes match the following. So vitamin A to its deficiency disease. So vitamin A is night blindness, A3. Okay, that is found in option 2. So option 2 is the right answer. Nowhere A3 is found. Vitamin K excessive bleeding. Deficiency can lead to excessive bleeding. Vitamin C will result in scurvy, and vitamin D will result in rickets. So the answer is option 2. Again match the following. Leucine. Leucine is what type of amino acid? It is a non-polar aliphatic R group, and it has got an aliphatic R group. So A3. Okay, A3 is found everywhere except in 2. Then serine. Serine is an negatively, no, phosphorylated by kinase. Yes, serine, if it is present in the active site of an enzyme, it can be phosphorylated by a kinase group. So 1B. Then glutamate is negatively charged R group. It has got an R group which is negatively charged, COOH. Okay, then tryptophan. Tryptophan is an aromatic R group. So the answer here is A3B1C2. Okay, that's in option 4. A3B1C2D4. Then next one. VLDL. VLDL is transport of endogenously synthesized triacyl glycerols, A4. So that A4 is found only in option 2. That's your answer. Then B, chylomicrons. B3, movement of dietary triacyl glycerol to various tissues. HDL is transport cholesterol from peripheral tissues to the liver, and LDL transport cholesterol from liver to other tissues. So the answer here is option 2. A4, B3, C1, D2. Then next one. DNA methylation. DNA methylation, it is for gene silencing. A2. A2 is found only in option 4. We will see others also. B3. Histone acetylation. Histone acetylation occurring on lysine residues of histones. That's correct. ATP dependent chromatin remodeling. It increases accessibility of DNA sequence. Nucleosome. Nucleosome formation, it is a barrier for transcription, because it is somewhat compactly arranged DNA molecule bound to histones. So if you have to give nucleosome access to transcription, you have to remove the DNA binding to the histones. So D4. Okay, next one. DNA polymerase beta. DNA polymerase beta is involved in DNA repair. A2. So 2A. Answer is option 2. B4. Leading and lagging strand of DNA synthesis. That is DNA polymerase delta. Then DNA polymerase alpha is a primase. DNA polymerase gamma is for mitochondrial DNA replication. So answer is option 2. Then match list 1 with list 2. Cyclin D, CD4, CDK4. So if you see the graph, you can see that cyclin D is at G2 to M phase transition, it is maximum. Sorry, G1 checkpoint. Cyclin D is for, that is here. Cyclin D is here, it is found throughout. So G1 checkpoint, cyclin D, A4. Cyclin E. Cyclin E is maximum at S phase. That is over here. So initiation of DNA synthesis B3. Then cyclin B. Cyclin B is here. So that will be transition from G2 to M phase. So next G2 to M phase transition cyclin B. Then P53 it triggers cell death D1. So the answer here is option 4 A4 B3 C2 and D1. Then next one, glycogen, cell wall of plants. Okay, where it is found. So glycogen is a carbohydrate storage in animals. A1. Sorry, A2. Carbohydrate storage in animal is 2. So answer here will be option 1, A2. Then starch. Starch will be carbohydrate storage in plants. C. Cellulose is found in the cell wall of plants and chitin is found in the cell wall of fungi. So the answer here is option 1. So thank you for watching my channel. Please do like, share and subscribe. I have uploaded still previous year question papers of CUET MSc entrance exam, and you can refer to my channel playlist for the same. Thank you.